For the paper we chose, the title is The Regulation of Actin-Myosin Interaction by Conserved Periodic Sites of Tropomyosin, presented by Jack McMonagle and Jake Shaulis.
So some relevant background information to fully understand this paper are understanding actin and myosin. They're both found in nearly all eukaryotic cells. They are responsible for a variety of cellular functions, including muscle contraction, cell motility, adhesion, cytokinesis, and organelle transport. If you look in this diagram on the right, we have a myofilament, and we see that actin composes the thin filaments, while myosin composes the thick filaments. Their interaction is best characterized in skeletal muscle contraction, in which they undergo a calcium-dependent mechanism with tropomyosin and troponin. More about tropomyosin. It is a universal regulator of the actin cytoskeleton. It has a two-chained alpha helical structure, and it can either activate or inhibit actomyosin. In doing this, it assumes three positions on the active filament. First one is the off position in the presence of troponin and in the absence of calcium, in which tropomyosin will block the interaction of myosin with actin. In the closed position, found in the presence of troponin and calcium, tropomyosin will slightly shift on actin to partially expose the myosin binding site and allow for some weak interactions. And then in the on state, which is in the presence of the myosin heads, tropomyosin will further shift to allow for some stronger myosin binding and thus muscle contraction.
The central question of this study is which residues of tropomyosin are required for cooperative regulation of actin-myosin interactions, and how do these residues structurally contribute to the transition of actin-tropomyosin filaments from the inhibitory closed state to the activated state. And then these are how the researchers obtain the materials required for the study. So for methods, first they perform site-directed mutagenesis, which essentially they use PCR-based mutagenesis to substitute conserved surface residues and to find periodic repeats on tropomyosin. This allowed them to test their functional roles in the actin-myosin regulation. And then they performed an in vitro motility assay in which they used fluorescently labeled actin filaments where reconstituted with tropomyosin and calcium and troponin, and their sliding velocities over myosin-coated surfaces were measured. Next, they performed kinetic assays where they used biochemical kinetics were used to evaluate how mutations influence transitions between block, closed, and open states. And then a fluorescent probe study in which tropomyosin and actin were labeled with environment-sensitive probes to monitor conformational changes. Then evolutionary sequence analysis in which they compared sequence alignments of tropomyosins from multiple species to identify evolutionary conserved residues. And then finally, molecular modeling. They used structural models of actin, tropomyosin, and myosin complexes to visualize how conserved residues might form. This provided a structural framework for interpreting mutational and functional data.
So why is this important? I said before that tropomyosin is a universal regulator of actin, so this applies to a variety of cell types. This is not just constrained to muscle cells. It's also implicated in a variety of cellular functions. We're not just talking about muscle contraction here. So overall, the paper serves to provide answers to this long-standing question of how exactly molecularly does tropomyosin regulate the actomyosin complex.
For our first figure, so this figure was created using two different methods: the evolutionary sequence analysis, in which they used previous information from other studies to choose which residues would be mutated, and then utilized site-directed mutagenesis to engineer mutant tropomyosins. So as you can see here, this is an amino acid sequence, and in panel A, the tropomyosin mutations are present in conserved surface residues, which is rat-striated alpha tropomyosin, which shows mutations in the B, C, and F locations, and they were mutated into alanine in P2 through P6. And then panel B is a visualization of these mutations in a 3D model. The big takeaway from this figure is that conserved surface residues of tropomyosin were identified and systematically mutated within its periodic repeats.
So, figure 2, this is a motility assay. It's essentially measuring the sliding speed of the filament under 3 conditions. Our conditions are no activator, which is shown in black. We have NEMS1, which is a strong binding myosin head, that is in white. And we have our troponin and calcium, which is shown in gray. So in panel A, we see that the wild type has a high speed with NEMS1 and troponin and calcium, which is expected. The second half repeat mutants show a strong inhibition to their sliding speed, and the first half repeat control mutants show a similar speed to the wild type as expected. In panel B, we see that the wild type speed maxes out around a 200 nanomolar concentration of NEMS1. We also see that the P32 EDKA mutant cannot really be activated at any concentration of NEMS1. And just to clarify with that notation there, the P3 clarifies the period, so this is the third period. The 2 means the second half, and then the EDKE is our amino acid sequence, so in this case it would be glutamate, aspartate, lysine, and glutamate. And we see that other mutants show a pretty decent or similar speed to the wild type. And in panel C, we see that relatively all the mutants show an impaired speed relative to the wild type. So the big take-home message here is that these conserved second half residues in P3 through P6 are essential for activation and this optimal sliding speed of the thin filaments. And even with troponin and calcium, the mutants, for the most part, and I say for the most part because some of them do reach pretty high speeds, but for the most part they do not reach full speed, and so these residues are essential for the transition from the closed to the open state.
In figure 3, they use an in vitro motility assay, which measured the sliding velocities of actin filaments with wild type or mutant tropomyosins. In panel A, you can see the dependence of K-fast on actin or actin tropomyosin concentration. And in panel B, you can see the dependence of the K-slow on actin or actin tropomyosin concentration. And then they use these equations in panel C to determine the data here. And as you can see, the second half conserved residues of tropomyosin are required for cooperative activation of actin-myosin motility.
So figure 4 is a titration of the actin filaments, which are PIA labeled, and PIA is a fluorescent probe, so basically when it's interacted with, it releases some light that can be detected. It's titrated with the myosin S1, which is the myosin head. Our x-axis shows the fraction of S1 bound to the actin tropomyosin complex, and our y-axis measures that fluorescent conformational change in tropomyosin when S1 binds. So our wild type is shown in the circles here. It has a strong cooperativity. One myosin can open about 6 or 7 actin tropomyosin complexes. Our P32 EKDA mutant, which is shown in the squares, has a reduced cooperativity. It interacts with about 2 actin tropomyosin complexes per myosin head. The P62KEE mutant, which is in the triangles, shows a similar cooperativity to the wild type, but overall we see that these conserved residues are essential for cooperativity in the myosin-actin tropomyosin complex. Figure 5 is a molecular model of actin, myosin, and tropomyosin in the open state. They use cryo-EM to obtain these structures. Actin is shown in green, myosin is in red. Tropomyosin in the EM structure is shown in gray, and the crystal structures of tropomyosin positioned in the EM structure are shown in blue. The side chains of the mutated tropomyosin residues are shown in white, and they have blue labels. In panel A, we see few interactions in P3 and P4 with actin and myosin, but in panels B and C, we see more optimized electrostatic interactions of mutated tropomyosin residues with actin and myosin and P3 and P4 and P5 and P6. overall, this shows structural evidence for how these conserved residues of tropomyosin enable activation of the actin filament.
So this table was provided by the authors in the paper, and this was used to support the data collected in Figure 2 and Figure 3. So what can we take away from all of this? So what we think are three main takeaways that the authors found from all the research done for this study. First of all, the authors found that conserved periodically distributed surface residues of tropomyosin are essential for the regulation of the actin-myosin interactions. They also concluded that residues in the second halves of specific repeats, like period 3 through 6, are crucial for the transition of thin filaments from the closed to open state, even with the presence of troponin and calcium. And then finally, as we saw in the cryo-EM model, structural modeling further suggested that these conserved residues may form electrostatic contacts with actin and myosin, which in turn creates a mechanism for tropomyosin to regulate actomyosin across muscle and non-muscle cells.
So, how can we apply this and what questions remain? Well, tropomyosin mutations are implicated in some pathologies, including hypertrophic cardiomyopathy and some skeletal muscle myopathies. With this research, we now have further understanding into diagnosing or locating these mutations and possibly treating them. Tropomyosin is also found in many cellular systems that are not necessarily muscular, and so we now have a further understanding into how we can control these cellular functions, for example, motility and division. Some questions that remain: what other tropomyosin mutations inhibit or contribute to the transition to the open state? We looked at a select few tropomyosin mutations here, but there could be others that could inhibit or contribute to this function. And in these cases where maybe we do have some pathology, what pharmacological interventions may we be able to use? How might they interact with these mutations to cure these pathologies?